My name is Abla Creasey. I'm a veteran biotech and pharma person, and my last training was at CERM. I was there for 9 years and ran the clinical group and translation group. I'm the moderator for this session, and I just want to remind everyone that the area of CAR T has been on everybody's mind, mainly for the opportunity to even expand it. I just want you to know that for those of you who are not familiar with CAR Ts, we will define what CAR Ts are. Dr. Yang will do that. There are currently 7 approved CAR T therapies for hematological cancers. You heard someone say blood cancers. There are 3 approved indications: acute lymphoblastic leukemia, diffuse large B-cell lymphoma, and multiple myeloma was the most recent. So the question that we're trying to raise here is not to deal with hematological cancers, but can CAR Ts be applied for treatment of solid cancers or autoimmunity? And for that reason, we've invited the experts that are on this table, this roundtable. So they are going to answer some questions regarding the promise of CAR Ts for those indications. And I'm going to ask them to introduce themselves. So we start with Dr. Lili, Professor Lili. Go ahead, please.
Thank you, Abla. It has been a great pleasure, to join you and the others in this wonderful panel. I'm a professor of microbiology, immunology, and molecular genetics at UCLA. My research will focus on the translational research. We develop gene and cell-based immunotherapy, and including the CAR T therapy.
Thank you.
Thank you. I'm Jim Robinson. I'm the CEO of A2 Biotherapeutics. We're right down the road in Agora Hills, California. Beautiful Agora Hills, California. Woo! Shout out. Our focus is on cell therapy, specifically in solid tumors. So looking forward to the panel discussion today.
Thanks very much for the invitation to participate in today's panel. My name is Zach Roberts. I'm a physician scientist. I'm the Executive Vice President and Chief Medical Officer at Allogene Therapeutics. We are a CAR T cell therapy company developing allogeneic cell therapies for blood and solid tumors, as well as autoimmune disease.
All right. And I'm David Barrett. I'm Vice President of Cell Biology at Kite Pharma. I'm also a physician scientist, and we are developing CAR T cells for all kinds of indications, including heme malignancies, solid cancers, and autoimmune disease.
Thank you all. So we'll start with Dr. Yang. So would you please give us a succinct definition for what CAR T means so the audience who doesn't know what CAR T is?
Of course, yeah. So CAR indicates chimeric antigen receptor. So the whole concept of the CAR T cell therapy actually is an engineering model. It stems from nature and immunology, but it goes beyond the nature. Why? Because in the immune system, usually there are 2 ways of recognize, a target cell, right? Something out of the surface or something like internally in that cell. And immune system has 2, different kind of receptor system doing that. The antibody-based recognition, which recognize outside 3D structure, the molecules sticking over there, and then there's internal recognition, called the T cell receptor, recognize the breakdown of the internal pieces and then present it on the surface. So the CAR idea, the chimeric antigen receptor, is taking advantage of both systems. So it really takes advantage of this recognition of the surface biomarker with a very high affinity antibody, but to bridge that antibody recognition piece to a T cell receptor signaling domain. As a result, the chimeric antigen receptor, CAR, if you put that onto a T cell carrier, will be able to recognize the surface biomarker. You can define, that to be specific for a blood cancer, solid tumor, or the autoimmune related, biomarkers, and then it will work to do its job. And so that's the idea about CAR T, but the recognition is higher affinity, compared to the conventional, T cell receptor. And also the design allow you to incorporate many helper functions from the T cell receptor. So it combines the major signal 1, which is necessary to trigger the interaction, and signal 2, which is necessary, called the helper co-receptor. You can integrate the helper signaling domain into the same single molecule. Again, as a result, the chimeric antigen receptor, now is able to allow a T cell to recognize a defined biomarker on a target disease with high affinity recognition, and also can respond independent of the necessity of both the receptor recognition and the co-receptor recognition. Everything is integrated into one molecule. So together, yeah.
Thank you, Lili. Thank you. So we go to David Barrett. Oh, that question for you is what's the secret for the success of the CAR-Ts in the hematological cancers?
Thank you. And I think the simplest answer is decades of dogged research, trying to understand how T cells work normally, trying to understand how you can effectively keep them alive in the lab, which is a big problem. If I take T cells out of the body, they're actually very unhappy. And you have to figure out how to keep them happy while you make the modification with the CAR and then give them back to patients. As Lili was talking about, one of the key things is what do you send them after? What molecule on the surface? And in hematologic malignancies, there are normal molecules on the surface of cancer cells. The technical names are CD19 and BCMA. And they're on normal B cells as well in your body, as well as the B cell cancers, the lymphoma, leukemia, and myeloma. But the good news is you can live without normal B cells. You can do just fine, carry on with your life. You won't really miss them as long as you're under the care of a good doctor who's monitoring you. But the big key there is that you could send these very powerful CAR T cells after the B cell cancers and not have to worry about the off-tumor effect. In addition, T cells live in the blood and lymphatic system, which is where leukemia and lymphoma lives. We didn't have to do anything special to get the T cells to find the cancer when we gave them back to patients. So there are a number of natural advantages, both to the way that we made CAR T cells, but also to the hematologic diseases that made them more likely to be successful. And this has been borne out now with 7 approved products and incredible clinical responses, better than any other kinds of therapeutic modalities. So that's a very exciting, very powerful thing. But how do you translate those natural advantages to tougher situations? What happens if your cancer doesn't live in the blood normally? It's in the liver, it's in the brain, it's a breast cancer or something else. How do you make sure the CAR T cells know where to go? And also, how do you do it safely? Is there a safe target? Again, the B cell malignancy cancers, those targets were safe because the normal cells that they were on, you can live without. You can't live without the rest of your brain. So if you're targeting a brain cancer, you better be sure you're targeting it with something that is only on the tumor and not on normal tissues. And that's been one of our great challenges, which we'll talk about, I think, more in the session. But certainly, we had success early in hematologic cancers because there were a lot of natural, the term low-hanging fruit has been used, but lots of natural advantages to taking a T cell and saying, what do you normally do? Where do you normally go? And matching that up with a target that was safe to put it after. Because one of the most intricate things about your immune system is it knows who you are and it knows what's not you. And when we make a CAR, we are breaking that intentionally to direct those immune cells against a tumor that the immune system otherwise wouldn't see. So we have to be very careful as the scientists and engineers and physicians who do that when we break that system to intentionally send it after a tumor, we're doing so safely.
Thank you, David. So we go to Jim. Has the CAR T therapy been tested for solid tumors? Tell us more about it.
And I'll use A2 as an example, if that's okay. So we have had 2 programs in the clinic, 2 autologous programs focused on solid tumors. First program targeting CEA, the second one targeting mesothelin. So we have dosed over 21 patients to date in both of those programs. And I think the point that David was making right now is the key point. Is as you think about solid tumors, it's been more difficult, and part of it is starting with the first principle of making sure you could target what you want to target and selectively kill that and spare the healthy cells. And that's the first principle that we had to really focus on. And we've developed what we call our TMOD platform, which is logic gated to be able to really identify the cancer cell and spare the healthy cell. And it's one thing to do it preclinically. It's something entirely different to do it in the clinic. And so as you go in the clinic, you're looking for safety first and assessing every patient as you dose them to determine whether or not there's a safety risk or safety signal that you need to be really monitoring and or learning from. The good news is we've dosed over 20 patients, as I said, but we have not had any dose-limiting toxicities yet. We've seen really strong anti-tumor activity, and we've had 2 partial responses so far. So we believe on the path of figuring out how best to treat solid tumors on a broader scale than I think historically we've been able to do.
Will you please give us examples of solid tumors that you've tested?
Sure. So colorectal, pancreatic, lung, ovarian, those are just a select few of the tumors we've been focused on in terms of our clinical development programs.
Okay. Zach, do you want to comment on that too?
Yeah, so another solid tumor where I think some of the exciting data that Allogene is about to present and has been presenting now for a couple of years in our phase 1 program is in kidney cancer. And so this is an area of high unmet medical need. Patients, as of the last decade or so, have had a number of therapies that are efficacious in this setting. However, almost invariably, these patients eventually develop resistance to each approved medication and therefore are left with metastatic and progressive disease that is untreatable. And many, sadly, these patients succumb to their illness, just like in other solid tumor settings or blood cancers, for that matter, that end up being refractory. But one of our products, Allo316, it's an off-the-shelf allogeneic cell therapy. We're just about to share some data at ASCO, but as recently as last November, when we shared data at SITC, we showed about a 33% response rate in patients with multiply relapsed refractory metastatic kidney cancer. And just to add a little bit, another piece of what makes solid tumors more challenging than blood cancers, it's not just their availability in the blood, but it's to a large degree the heterogeneity of these tumors. So unlike blood cancers like lymphoma or myeloma or leukemia, where the cells largely look the same, certainly as it pertains to the target of the chimeric antigen receptor, in the case of solid tumors, for quite some time now, we've known that no 2 cells are really alike. And so it makes it much harder to find a single target that is applicable to all cells in the body. Another major confounder related to the availability of these tumors and access by the CAR T is the ability of these cells to actually burrow into these areas, these oftentimes very large foci of tumors in the body. And to bring it back to Allo316, we have evidence now showing that these cells, the Allo316 cells in this kidney cancer setting, actually are getting into that tissue and killing tumor in situ. So this is kind of one of those things that we need to engineer our way into solving this problem to really bring the promise of cell therapy to solid tumors.
Along those lines, Zach, did you have to modify the CAR T for the solid tumors?
So the short answer is yes. We had to another piece of this puzzle that differs from solid tumors and liquid tumors is liquid tumors. Often you can induce deep remissions within weeks, 2 weeks, 4 weeks. In fact, we just saw this morning in the morning session a patient with follicular lymphoma who had a very daunting PET scan at the time of her infusion, but then 1 month later had a completely clean PET scan. That almost never happens in solid tumors. So you need to engineer a cell therapy that is able to persist in the bloodstream long enough to whittle away at this tumor, which often can be kilograms of tumor. So in the case of Allo316, we have a part of the engineering is something that we've nicknamed the dagger effect, which enables these CAR T cells to persist for quite a bit longer in the bloodstream than certainly a typical allogeneic cells, which generally gets rejected very quickly. So we've overcome that short persistence window by engineering the cells to last longer so that they can kill more tumor cells and ideally put patients into a durable remission.
Great. And David, you wanted to say something?
Yeah, just, to carry on with the theme of the breadth of solid tumors, at Kite, we are also presenting at ASCO results in glioblastoma, a deadly brain tumor, where we have a dual antigen targeted CAR. So again, to Zach's point, the heterogeneity, not every tumor cell is the same. And so a CAR that has been designed to go after more than one target, and in addition, giving it directly into where the tumor is, being able to deliver it directly into the brain where the tumor lives, and seeing the incredible results and persistence in the brain as it works against those tumors. So the breadth of solid tumors that can be targeted is really also represented here. And, tackling those aspects of, you can't just get away with the simpler versions that have worked for hematologic malignancies. It is more difficult. You have more complex engineering to do. And we're starting to do that again with complex multi-gene targeted aspects that can really start to get at this solid tumor environment.
Thank you. So there are new publications about the potential of CAR-Ts being a very important therapy modality for autoimmunity. So we're going to switch from solid tumors to autoimmunity. So we'll start with Zach again.
Yeah, so this is an interesting development for the field. I remember I used to work at Kite, and we used to, obviously, we traffic in B cells and B cell tumors, but B cells are a key component of the autoimmune pathogenesis in many, if not close to all cases, all diseases. And so we used to speculate in the early days that this could be done for autoimmune disease, but it really took a pioneering group in Germany to run the first clinical trial. Now, 3 or 4 years ago, they launched it. And that really did change the field quite a bit overnight. And so there are now several different therapies targeting predominantly CD19, the same target that we go after with drugs like Yescarta for B-cell malignancies, showing that a single infusion of these CAR T cells can induce drug-free remissions that last months or even years in patients that had treatment-refractory lupus and myositis and systemic sclerosis and myasthenia gravis. I mean, the list is ever lengthening. We're at the beginning of this story, I think, and just as we've seen, even in this brief conversation, the need for enhanced engineering and more thoughtful approaches to tackling what on the surface may seem a fairly straightforward problem is probably going to be needed. And so one of the areas that, again, Allogene is looking into is and taking a page out of David's point about targeting not one, but 2 different antigens to try to enhance the efficacy and the feasibility of this therapy. So we've got a product that we're about to launch clinical trials within just a short while, where we're targeting CD19 and CD70. And CD70 is an interesting marker. It's the target of Allo316 in kidney cancer, but it's also a really interesting molecule because it's also expressed on many of the immune cells that cause autoimmune disease. So we're really attacking the autoimmune pathology from both angles, the CD19 on the B cells and then CD70 on T cells as well as B cells. And hopefully we'll be able to use some of this dagger effect that I referred to a moment ago to really engender prolonged persistence, at least long enough to induce an immune reset. So this is a really interesting new area for CAR-T, and I hope that we get to participate collectively in this field for the next many years and really address an area of very high unmet medical need.
And as far as re-engineering the T cells or the CAR-Ts, is that more extensive or just, like you mentioned, a couple of antigens?
Well, it's a very interesting question. I mean, there are many ways to approach this. I mean, in our case, we've decided to do relatively straightforward gene engineering. Again, we're an off-the-shelf product, so we're using healthy donor to give our starting material to make our CAR T cells. That differs from an autologous platform, which draws cells from the patient and starts with. So we have to do some enhanced engineering to prevent premature rejection of our cells and also prevent our cells from inducing a reaction called graft versus host disease, which we would expect when we give a healthy donor's immune system to a patient. So we've taken a fairly parsimonious approach to engineering our cells, targeting the CD19 and the CD70. But there are a host of other strategies that other developers are doing to cloak their immune cells, to make them do different things in the patients that is slightly different in autoimmune patients than it is in cancer patients.
Thank you. David?
Yeah, to add on to that, I think a key aspect is you can use cells from the patient, or you can take an off-the-shelf approach. But also to remember that in autoimmunity, the goal is slightly different. In cancer, it's very much so you want to kill every last cancer cell. You need to do that. You have to do that. With the approach of CAR T cells in autoimmune disease, you want to eliminate the diseased B cells for as little extra toxicity as you can handle. The least amount of other therapy, like the preparative lymphodepletion regimen, the least amount of other things you can get away with, because you don't need to kill every last B cell in the patient. You need to get to the ones that are causing their lupus or their myasthenia, and you want them to recover very, very quickly. So there's a slightly different tolerance for I think the toxicity and the other aspects when you're trying to go for autoimmune disease. And that's something that we all wrestle with is, as again, Kite is taking an autologous approach, also targeting 2 antigens. It turns out that a lot of us think the same way about the same problems. Ultimately there will be many solutions to this problem, and we'll all learn from each other. And I'm as a scientist, very excited about that. And as a clinician, very excited for the patients. But yeah, it is definitely a little bit of a different calculation as to how you try to thread the needle for the most patient benefit.
Is there potential for an allogeneic strategy there too?
Of course. And I mean, as mentioned, because again, you have the persistence issue that Zach has talked about with allogeneic products. That is a challenge. But in autoimmune disease, as the Dr. Schett and the German data showed, you don't need it to last for as long as it seems to last in leukemia patients, for instance. So we don't know exactly what that window is, but it does open up room for lots of allogeneic and autologous approaches to tackle, and again, maybe different needs for different diseases.
Thank you. Jim, you want to comment?
I think it's been said already. I think the key here is, can you selectively target the cells you're trying to either kill or rid from the body and spare the healthy cells? And so that's, again, the premise of our entire TMOD platform, is the ability to do that. We have actually worked on autoimmune, specifically MS, preclinically. So we have the ability to move forward if we want, but right now our focus has been predominantly on solid tumors.
Thank you. So Lili, you want to comment on what challenges does this field face in terms of CAR Ts, whether it's side effects or manufacturing or whatever? So please go ahead.
Certainly. Yeah, I think the panel already touched, a kind of various couple of aspects about it. One is the manufacturing, right? So the allogeneic off-the-shelf option obviously can offer a lot of advantage for manufacturing, for delivery, for price-wise. And the other limitation is for the solid tumor, like beyond the heterogeneity of the tumor cell, which you can try to tackle, by target multiple, the biomarkers. And there's also a microenvironment challenge over there. Like solid tumor has this extremely immunosuppressive microenvironment. It's comprised by this immunosuppressive immune cells and the environment, metabolites. This whole, it's a challenging, field. Like in order to really make the anti-solid tumor, CAR T cell therapy really, powerful, that those aspects need to be encountered too. And Abla, you asked about the additional engineering approach. I think those are, touched by that. Like you can block the TGF-beta, you can kind of provide some other, strategy to deplete the immunosuppressive molecules and the immune cells, like in the microenvironment.
Thank you. So we need to address part of what the regulators constantly worry about is the consistency of producing those kind of products. So maybe Zach or David, you can comment on it.
Sure. I think that is an inherent challenge from the autologous CAR T cell space, right? When I'm getting the starting material from every patient is their product. Every product is unique to that patient. So how do you ensure consistency about what you're trying to deliver? And it's incredibly challenging. And I think a big part of the journey of cell therapy is with the regulatory environment. These are gene-modified T cells or genetically edited T cells, and those carry with it incredible responsibility to follow patients for safety, to identify safety events and things that take place. But it has really been true that you have to try to come up with ways to say, all right, what I get from each patient I cannot control. But what I can do is I can control exactly how I treat every patient's T cells. I can show the range of what it looks like, how many CAR molecules were on your cell, how well did they grow over this period of time. Did we do all of the same safety checks on every patient's cells and every ingredient that went into that? And we can do that. And what you can see over treating more than 20,000 patients, actually, my boss is probably going to think it's over 25,000 now. I got to get that number right. But treating tens of thousands of patients, you can see this diversity, but you can also see the consistency within it and the evolution of that. And I'm incredibly proud of our manufacturing team for delivering that kind of consistency, starting with variation. But one of the things you can do, if you're an allogeneic product, for instance, is you can start with select healthy donors and get potentially even more consistency with the trade-offs that go with the extra engineering you have to do.
Zach, you?
I mean, yeah, I think Dave gave a great answer, and the consistency of manufacturing, we get 100 or more doses from a single manufacturing run as opposed to 1 or 2 doses, which is typically what you would get from an autologous manufacturing run. And even that number, 100 or more, can probably swell quite significantly with manufacturing improvements. But to David's point, I mean, this is really, the process is the product. It's a little bit of a cliche these days, but it's true in cell therapy, because how you modify the cells, whether they come from the patient or come from a healthy donor, it is exquisitely well controlled from one manufacturing run to the next. In an allogeneic platform, you get 100 or 1,000 doses, let's say, from a single run, but the idea is still the same. Of course, there are other benefits from that kind of manufacturing scale. Theoretically, the cost of goods goes down quite substantially. That does sort of enable additional access, which is another key barrier in CAR T-cell therapy. So I think there are gives and takes for each of the different platforms, but I think allogeneic does bring a certain element of scale and access that I think is unique to it.
Go ahead, Jim, if you...
I mean, what I would add, I think one of the missing points is the people aspect. So we're very fortunate. We have a clinical stage manufacturing facility, both autologous and allogeneic. And so we're also very fortunate to have very experienced people that look at this as not only in terms of an end-to-end process that requires you to be precise and repeated time and time again, but also the willingness to continuously improve and look at the opportunities to refine the process, whether it's process development or whether it's what you're doing in manufacturing, and really have the spirit of learning on each and every opportunity, whether it's your patients you're looking at from an autologous perspective or allogeneic. So I think the key here is not just in terms of the process, but it's also the people you have that have the experience that actually can have the willingness to continually focus on improvements that can be required to continue to have the consistency that's necessary to meet the needs of your patients and the regulatory authorities as well.
Along those lines, there was a question about handling of the cells when they are before administration in the clinic, chain of command and management of that. Could any one of you comment on that?
Yeah, I think it's incumbent upon the sponsor to ensure that the training takes place at the institution and assume nothing. So when we dose a patient, we have our team there at the institution that's dosing the patient, make sure that we can manage the process from beginning to end, including assisting in the principal investigator on any issues that might arise. So really making sure that we have the process tight, which we do, but also making sure that we're not taking anything for granted and we're on site when these patients are being dosed is, I think, our key to our success so far. And it's probably not unique to what we're doing.
I'll add to that. I think, when I used to work at Kite, I used to spend an awful lot of time on transplant floors supporting the infusion of these clinical products to patients. And I think, and I know you did at Penn, probably even more so. And I think that's a critical piece of the early development of these novel agents. In the blood cancer world, they've been doing bone marrow transplants for decades. So you're able to leverage a lot of that infrastructure and training to administer those products in the early days. But I think generally now, certainly as we get into solid tumors, that expertise does not exist in a solid tumor oncologist, and it certainly doesn't exist in a rheumatologist. And so, again, as we're consistently pushing these boundaries and expanding access to cell therapy to areas where there isn't an existing infrastructure to support them, we have to be careful during the development. But I'll say on the other side of that coin that we also have to find a way to make this more straightforward for clinicians and for patients. And the closer that we can get to a pharmaceutical distribution and administration model, the better, the more access we're going to have, the easier it's going to be for doctors and nurses in the community setting, for example, to administer these products. And ultimately, if we're developing products that require so much specialized expertise to administer safely, we're sort of building ourselves a set of limitations that is going to, frankly, hinder the opportunity of these products to improve outcomes.
Yeah, just one final thing to add to that, which I think is a nuance to this aspect, because we do agree, all of us, I think, that access is a huge issue for cell therapies. Where cell therapies started was in huge academic centers, quaternary transplant centers like Penn and Baylor and Seattle and Sloan Kettering and all the NIH, right? I can infuse a cell therapy product in a trailer in a county in rural Virginia. I can't take care of what happens to that patient there because of the nature of the side effects that we have to anticipate with current generation cell therapies. Cytokine release syndrome, immune effector cell associated neurotoxicity syndrome. These are things that require immense supportive care. So there are complexities to the, as we say, the chain of custody, but that we're getting much better at as we've stood up at Kite more than 190 advanced treatment centers where we do the training, right? Here's how you prepare the cells to send to us. Here's what you can expect when you get the cells back. Here's how you process them to infuse. And that part typically goes very well. But again, it's that infrastructure of what do I expect? When do I expect it? How will I care for my patient? And in addition to that, can I give it in my community hospital so my patient doesn't have to travel 60 miles and figure out who's going to watch their dog or take their kid to school while they're at 60 miles away for 2 weeks. So there's an incumbency on us to make safer products that are simpler to give to work on that access issue while increasing and in providing the expertise of our people to those centers to make sure that that can happen. So there are many aspects to this which all of us are trying to solve, because each one of us that solves a problem means more patients get access to therapies that help them.
Thank you, David. There's still the question of failure rate of lots. And it's not always guaranteed that every person can get a CAR T therapy. Can you comment on that?
Specifically with regard to failure rates, right? Like I'm getting material from a patient with cancer or lupus or glioblastoma or pancreatic cancer or kidney cancer. Those cells have been exposed to chemotherapy or other therapeutic agents. That person has cancer, which inevitably means that something was wrong with their immune system in the first place to help enable that cancer to appear. What do I do if when I try to make those cells into CAR T cells in our facility, they don't work? What if they're broken and they fail? They don't grow. They don't express the CAR. That is something that we've learned a lot about from translational data, learning from those patients that their products didn't work. Why? What was broken? How can we look for that? How can we treat the patient differently, get their T cells earlier? I'm keep looking at Zach and I keep giving credit. How can we use healthy donor T cells that don't have some of those problems? We continually learn and improve on that. I can say at Kite, our manufacturing success rate is incredibly high. It is in excess of 96%. I can't see anything because the lights are in my eyes, but I'm sure a regulatory colleague is going to point at me and tell me I'm too low. But it is incredibly high, and it comes from that experience and consistency. And we learn now much better how to avoid that. But to the extent that when you're dealing with a personalized medicine product, there are still things you don't know about each individual patient. But the ability to learn and make that happen less and less often, shorten the time that it takes. Again, Kite has a turnaround time of 12 days now. And so we can get the cells back to patients really quickly with a high success rate. But we've also done 25,000 plus patients. That takes a while to get that smoothness up, get those quality people that we talk about to make that delivery.
Thank you, Jim.
Yeah, I think the benefit of where we're at, we have been able to learn from the experience of Kite and Gilead and companies before us. And I think one of the points I'll build on is how quickly can you, when a patient is diagnosed with cancer, can you actually get their T cells? And so the earlier in their disease, the better, we believe, in terms of having a ability to grow the cells to the extent you need to be able to create a treatment. So we've been able to really leverage what we've built, which we call Basecamp, which is our ability that the moment we can get a patient identified at our sites, get them consented, we apheresis them. So we have their blood, we have in the freezer. So when they advance, we're ready to go. And so that also cuts down on time in terms of being able to, they've progressed, how long will it take you to manufacture their cells, how long will it take you to infuse them. So lessons learned from companies up here on the stage have been really important to us. And so we've leveraged that learning. And I think because of that, I'd encourage the learnings to continue to be shared, because it's only going to make us all better. But clearly a key point is making sure you can capture the T cells as early as possible.
Thank you. What's often on the mind of potentially everyone in the room and out there is that the cost of this therapy, and who can afford it, how long, and how are we going to reduce it? So maybe access affordability is something that was mentioned earlier, but the team would like to address some of that as well. So David, you want to start?
Sure. So cost is an issue, but I think it's also, and this is something that I think a lot about as a former pediatric oncologist and bone marrow transplant physician, cost over time for what benefit? When I think about children with spinal muscular atrophy, which in its severe form is completely incurable and results in a child who is progressively disabled until they die in adolescence. If I have a gene therapy that could cure that when they're a baby, and they don't have 15 years of increasingly intense care that ends up in the end of their lives as total care, as children who are paralyzed, who cannot eat or move or do anything on their own before they succumb to their disease, the cost of that care over time is immense. And do I pay for a gene therapy up front that saves that cost and also results in a normal, healthy child who gets to grow up and have a life? And in some ways, some of our therapies are set like that as well. The cost of cell therapies right now is incredibly expensive as a one-time upfront payment. But the promise of cell therapy is that that's the only treatment you'll need, and that you will achieve in the majority of patients a cure of your hematologic malignancy. I think also of some of the other treatment models like bispecific antibodies or T-cell engagers. T-cell engagers can cost $10,000 a month. You can get it every month for a year and still have cancer at the end of the year. So where are we at this? Absolutely, the amount of upfront cost is an immense barrier. And we need to think about that as an industry. We need to think about that in terms of our healthcare as to how we can address better how to make therapies more affordable. Increasing the access is absolutely one part of that. But also, how do we rethink this aspect so that it's not so daunting in this high sticker shock upfront cost? There are also, again, the trade-offs of, as Zach has mentioned, he can make 100 doses of an allo product, for essentially a lot of the same materials, but how many doses do you need? What is the length of that effect? And how do we balance all of that out? And the holy grail, of course, is to achieve something that's incredibly cheap to make, that is incredibly fast to give, that gets you the results you get now from the expensive, labor-intensive autologous cell therapy process. We're working on that. All of us are. Everyone here is. Probably a lot of you out there are as well. But it is a challenge. But again, I think that the cost for the benefit is where the value can lie. And, for a product like Yescarta in second-line lymphoma, 50% 5-year overall survival, and this extended amount that is actually a cure for second-line lymphoma, versus an autologous transplant, which costs almost the same amount but does not result in that same level of cure. These are the arguments that we have to have, because it is a challenge for the whole industry. We don't want it to stop the development of new therapies. So
thank you, David. Thank you. So Jimmy, you have a minute.
Yeah, just this could be a whole other session for 4 hours, but I would say is the old school. If we're looking at value-based healthcare, right? What is the definition of that? Is cost over outcome equals value. If you look at the outcome that was David just pointed to in terms of the outcome a patient will get from multiple myeloma treatment from Yescarta, well, that's a pretty sizable outcome, not only for the patient, but also for the healthcare system. And so I would say that if you're looking at cost effectiveness based on outcome, I think cell therapy provides a real cost benefit.
Excellent words. Lili, you want to give the final words?
Sure. Yeah. So, follow up on the affordability, delivery, logistics, and the cost, at the end, or the manufacturing complexity, how to make it more consistent, maybe, other cell resource can be a consideration. Like, California Institute for Regenerative Medicine Center has been a wonderful funding agency, support the stem cell derived therapy. So ultimately, maybe, one day, like we can make the iPSC derived cell therapy, CAR T cells, and then that may address many of those challenges.
Thank you. We have 30 seconds left for one good question from the audience. Anybody? I can't see.
There's someone waving over there.
You're going to have to make it a good one, though. So I'll just repeat the question, everyone here. How do the current CAR T cell therapies compare with in vivo therapies or in vivo CAR T cell therapies? In vivo CAR T cell therapies don't have proven benefit yet. They are in early phase trials, none of which are in the U.S. I think we're all very interested in vivo therapies, and certainly all of us have thought about them. What if you could skip the manufacturing? What if you could skip taking the cells out of the patient and having to do anything else, and you could instead give some form of therapy that allowed it to, essentially the manufacturing to happen in the body? We have to stop because we're over, but I think that the thing is we still want to know, but we don't know yet.
Thank you. Thank you all for coming.